say, describe the structure of DNA. Structure of DNA. Guys, when talk about the structure of DNA, look at the characteristics of DNA. So I think that's done. The characteristic of DNA is double helix is made up of 2 strands. It's made up of the nucleotide. It's made up of nucleotide, yes, which are made up of phosphate, the oxyribose sugar, and nitrogenous bases. We talk about the nitrogenous bases, talk about all of that. Yes. Challenge. Don't go to the paper without knowing DNA replication. Yes, don't go to the paper without knowing translation. Don't go to the paper without knowing transcription. And then describe the description of the structure of DNA. So one will come. Won't tell you which one, but one will come. You'll come. We're going to discuss the paper after. You'll know. And then now, if they ask you a question that give the difference between DNA and RNA, that one is simple. You can talk about everything you know. Make sure that you draw the table. Don't leave it hanging like this. You want a complete table. You mentioning this, you writing like this, and you say DNA, and the other side you say RNA. Guys, put DNA, put RNA, close the table. If you don't know anything, at least you got a mark. Yes. If they ask you to tabulate, draw a table, put the title, close the table, leave. If you don't know anything, yes. If you know something, then write something. Yes. I'm done with that. But now the challenge comes in when they ask you that give the difference between DNA nucleotide, nucleotide, and RNA nucleotide. Here, you only look at the nucleotide of DNA, the nucleotide of DNA and the nucleotide of RNA. And there are only 2 differences in there. DNA has, don't talk about the phosphate because it's the same. DNA has deoxyribose sugar and the RNA has ribose sugar. And then DNA has thymine, RNA has uracil. Done. If they ask you about what? About the nucleotide. Is it clear? If they ask you the difference between DNA replication and transcription, you have to know the differences. DNA and transcription. We're going to spend a few minutes here. I'm only going to focus on the things which you fail. Which you fail is what I'm going to focus on. Yes, too much. Then we are saying that DNA, you talk about DNA, in this case, in DNA replication, 2 strands act as a template. The other side, you talk about 1 strand transcription, 1 strand act as a template. Guys, only know at least 3, but we want 2. In most cases, we want 2. We won't ask you more than 2, the differences. Don't go to the paper. If there's a lot, just know few things. Don't on each, at least go have a dose on each, whatever I'm going to talk about. Yes, then you'll be able to make it. Then we are saying that, yes. Then another thing which you go off is about the mutations on DNA. About the mutations on DNA. So when they ask you about the mutation, let me just use my book here so that you understand. Excel in Life Science Distinction Material. Here, let me just, I don't want the structures. Okay, I don't want the structures. What I want is this. What I want is at the end here. Yes, at the end, the mutations. Yes. Guys, let's answer this question. That what will happen if a mutation changes triplet number 1? Triplet number 1, it's this. From this, the same thing. From this to this, it means that this has been changed. Is it clear? You changed G to A. So now what you do when they ask you such a question, you first identify what is here. It is a codon. What is given here is DNA. Therefore, I have to convert this into messenger. Remember, we say that. Let me do like this. Yeah. Remember, we said that when you talk about code, it means DNA. DNA. When you talk about codon, it means messenger RNA. 3 bases on messenger RNA. When we talk about anti-codon, we mean 3 bases on transfer RNA. So now the question is asking, it means that it's changing from DNA to codon. It means that it's talking about messenger RNA. Is it clear? Yes. So now, what you do is to change this into messenger RNA. So change this into messenger RNA. So when you change into messenger RNA, this is going to be A goes with U, T goes with A, G goes with what? G goes with the C. So I'm going to CUA. I come here, look for C, C, CUA, C, no, AUC, AUC, where is it? US is triosine. So this is the triosine. UCA, UCA, isn't it? Then what about this? Now I change it again. Also is U. It's gonna be. It's gonna UAU. What does it mean? UAU is also triosine. So it means that a change from this to this, yes, will lead to this USC, from USC to UAU. That's the first mark. It will, you converting this into codons. You understand? Into codons. Is it clear? Which will code for the same amino acid, triosine. Therefore, there will be no change in the protein. Why? Because it's the same amino acid which is being coded for. Can I repeat it? I said, when they bring such a question, I know when they bring it, and I know it will be there. When they bring such a question, students lose these marks. Change in this, first find out, changing, because here it might be tRNA, and here is codon. So you change into the codon, which is messenger RNA. Is it clear? So change from ATG, basically result in this, and the change in this result into this. Is it clear? And these two, they code for the same amino acid. Therefore, it does not code for a different protein. It's like me calling you, let me check. Let me check here. It's like me calling you Lerato, Lo, Lailo, whatever. Ne? Sorry if I pronounced you wrongly. Ne? I can call you Lerato, or I can call you whatever, ne? The second name. I don't want to pronounce it badly. Ne? Yes. So if I call you the other way around, you say yes, sir. When I call you with another name, you also say yes, sir. You understand? So it means that they are coding for the same amino acid. However much the codes are different. Is it clear there? Yes. Then, what about base triplet number 4 from, yes, base triplet number 4. So this is the first instance. The second instance, what will happen to a protein if the base triplet number 4, ATA, is changed to ATT? So ATA, I have to convert it because the DNA, I need them to be in messenger RNA. ATA is going to be, T goes with A, U. And then this one is going to be U. So now I say that this will change into this. Okay, this will code for this, and this will code for this. Cool. That's point number 1. But what does this code for? AAU. AAU is valine. What about UUU? UUU, is it valine? No. Where is it? We have UUU. No, man. It's not UU. Yeah. What am I doing? It's AAA, ne? It's AAA because T goes with A, T goes with A. So it's AAA. So what am I looking for? Is the AA is proline. So it means that if you look here, this one code for valine. While this one code for proline. You see? So it means that these two, they are different. Is it clear? So it means that even the protein will change. It's like calling you Carabo, and then I call another, I call again Cabero. And Cabero is different from Carabo. You see? Yes. So it means that now this will code for this will code for this, of which this code is for valine, and then this, you write this in full, that AAA, ne, which code for proline. Therefore, a different protein will be formed because of the different amino acid. That's the scenario number 2. Scenario number 3. Yes, one scenario will come. Ne? Explain what will happen to a protein if DNA base triplet, or if DNA base has changed. Yes, there they have not talked about you have to interpret it automatically. If the DNA sequence has changed, one base has removed or has been replaced, whatever, it will lead to a different sequence of messenger, which will lead to a different sequence of transferrin A. Yes, which means a different sequence of amino acid. You understand? Hence a different protein. Different sequence of DNA will lead to a different sequence of messenger RNA, which leads to a different sequence of transferrin A with its amino acids, which leads to a different protein. That's a study scenario. The first scenario, it might not change the protein. Therefore, the protein is not going to change because the amino acids are being coded by different codes as we saw it. The second scenario, they can code for a different amino acid, meaning that valine and proline, for example, therefore a different protein. Or they can ask you what will happen if DNA base sequence changes or messenger RNA sequence changes. So you start from messenger RNA sequence. If it changes, it will lead to a different sequence of transferrin A with its amino acid, hence a different protein. Guys, don't go without knowing those things. Lastly, on DNA, it's this. Let me just use this example, but you still have questions. You still have questions we can use. Please, 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 don't go to the paper without knowing that. Someone is telling me they have given you T or C. Just mind A and C. Yeah. So guys, make sure that you guys get the things right. Yes. If you have any challenge, let me know. Yeah. Lucy. Lucia. Yes, you're saying adenine join. Yeah, I'd made a small mistake, but yeah, I saw it when I was trying to. Yeah, I'm also human. I can make a mistake. Yeah, but when I do it, sometimes when you talk, you can talk about something else, and then what in mind is something different. Yeah. All right, but I'm sober today. I'm sober. I don't drink and smoke. So I'm sober. All right. All right. Let's continue to the next. Guys, don't go without knowing how to label these things. What is this? It is amino acid. What is this? Is ribosome. What is this? What is this? It's transfer. You see, it is a T. Guys, check. It's a T. tRNA. If you don't know it, try to see if that T will come up. Check. You see? It comes out. It's a T. So it's a T transferrin. Guys, this is not a protein. It's a polypeptide chain. Protein is 50 amino acid and above. Is it clear? So this is DNA molecule. This is messenger RNA. This is nuclear pore. This is the nuclear membrane. Please go when you know how to label these simple things. Is it clear? Yes. This is the transcription. It occurs in the nucleus. This is translation. It occurs in the cytoplasm. Therefore, if they ask you what is this space, is cytoplasm, and what is this space, is the nucleus. Yes. Lastly, but not the least on DNA, so that we go to meiosis. Two things on here. The first thing is if DNA is 30%, if adenine is 30%, what is the ratio of guanine to cytosine? What's the ratio of guanine to thymine? Let me just say like that. Guys, you have to know how to do the base calculation. A goes with the T, C goes with the G. If this is 30%, automatically also this is 30%. So 30 plus 30 gives me 60. So remember, this is percentage. Percentage means 100. So 100 minus 60, it gives me 40%. So this 40% is for these two. So divide by 2, so it's going to be 20, it's going to be 20. That's not the answer. They're saying the ratio. So it's going to be G to T, whereby G, where's G? G is 20 and then T is 30. So I divide by 10, I divide by 10. So I get 2 here and then I get 3 here. You know what you're going to do? The first objective we're going to do, we're going to say that 3 to 2. Because when you saw 3 and 2, you just say that it's A. And the last one is going to be 2 to 3. because you don't want to read everything. Make sure that you read everything because there is most correct answer. I remember in paper 1 whereby they gave a correct answer halfway correct, and there is the most correct answer just down. You understand? We always try to trick you to see that, yeah, you are off. But I know since you are here, you won't be off. Ne? Yeah, we are here to kill the paper, not the paper to kill us. Ne? Yeah, tomorrow we will make it. We will make it. We'll make it. I know. All right. Yeah, if do you have any question, let me know. Let me know if you have any question. Let me know so that I smash that question without. That's number 1. That's number 1. The last thing which I need to talk about is about profiling. This profiling. Someone might say that profiling is easy, man. Yeah, I know that profiling is easy, man. But there is a time when they are talking about comparing the paternity test. Ne? Let me see if this question is about paternity test. Identifying the criminal, or what is this? Okay. Sample, DNA sample. Okay, let me just read it. Detective investigating the scene of broken window, suspected blood was found in the criminal, identified. No, it won't give me what I want. Blood. And it won't give me what I want. Okay, now let me design my question so that now you understand what I'm saying. If they say that 2 guys, they are crying, or they are saying that, okay, you have a boy, and then you have 2 guys, ne? And these guys, they are saying that it's each is saying that is the father of this boy. But now the mother doesn't know who, because this mother slept with these guys, ne? These guys are the same day, ne? You understand? So now we don't know who is the father of this boy. So what you need to do is, if this is the DNA profile of the boy, guys, listen until we end this, ne? Because sometimes this question, when it comes, you are off. Ne? Okay, if this is the profile of the child, yes, if the profile of the child, I know DNA profile is very easy, but once it comes to this, you lose the marks. Yes, you lose the marks. Okay, this is the boy. Ne? And then now you have the profile of the mother. You have the what? The profile of the what? Of the mother. You have the profile of the mother. Is it clear? Yes, you have the profile of the what? Of the mother there. And then now you have the first profile of the guy. Well, the first profile of the guy. You have the first profile of the first guy. You have the first profile of the first guy. Ne? Have the first profile of the first guy. Yes. And then you have the second profile of the second guy. You have the first, you have another profile of the second guy. You have another profile of the second guy. Yes. Yeah. So now they ask you, this is the mother, this is X, this is Y. Is it clear? Who is the father of this guy? And after that, they can ask you to explain, and they say 4 marks. Explain 4 marks. That's it. That's what I'm saying, that's where students go off when it comes to this. Listen carefully what you're supposed to say. Why did I divide with the 10 so that I get the simplest ratio? Ne? Yes, I divided by 10 so that I can get the simplest ratio. Yes. To simplify the ratio, yeah. Yes, correct. What you said is correct. I divided by 10 so that I can get the simplest ratio. Equilibrium, I will reach there, don't worry. So, guys, look here critically. What you need to do here is to first, because I know the mother. So take all the bars which correspond with the mother. So this corresponds with the mother. Yes. This bar corresponds with the mother. Yes. This bar corresponds with the mother. So it means that, let me just use a different color so that we see exactly. So this bar is out. This bar is out. This bar is out. So now the remaining bars, not that one. I'm saying this bar is out with the mother, this bar out with the mother, and then this bar is out with the mother. So the remaining bars, these remaining bars, this, and this, it must correspond with one of the guy. If there is one bar which is not corresponding, then that's not a father. Is it clear? Yes. Now I come here. I look for this. What does this one correspond with? This. If there is no one who is corresponding with it, then it's out. You understand? Then is what? Is out. So this bar correspond with the with this, ne? Yes. So it corresponds with that. This one is a little bit down. And then this bar corresponds with that. This bar corresponds with this and this. So however much this one has one which is corresponding, but this one and this one did not match with it. So the remaining bars, you see that all the remaining bars, yes, have corresponded with this guy X. Therefore, guy X is the father. Explain your answer. Why are you saying that X is the father? X is the father because the bars of the boy correspond, the remaining bars which of the boy which do not correspond with the mother correspond with this guy X. All the remaining, the word all must be there. All the remaining bars of the boy, yes, after matching them with the mother, correspond with guy X. Is it clear? Because they always ask you this. Yes. And then students tend to go off. These bars must correspond with the mother. The remaining bars must correspond with the father. If there's one bar which is not corresponding, we will ask you, where did that boy get that gene? Understand? Remember, we get these chromosomes as a sperm and ovum. Where did that boy get that gene from? Are trying to mean that's a mutation? So that's why all of them must correspond with the father, so that we conclude that that gentleman X is the father of that boy. Yeah, guys, unless you have any question concerning about describe the process of translation according to the messenger RNA, the transfer RNA with the complementary anticodon match according to the messenger RNA, the transfer RNA with the complementary anticodon, yes, bring the required amino acid to the ribosome. The amino acid is joined together by a peptide bond to form a required protein. Guys, those are just processes I'm not going to go through. Maybe I can ask a question. What is DNA replication? When does it occur? When does it occur? When does it occur? It occurs during before the cell divides, during interphase. Where does it occur in the nucleus? Why does it occur to double the chromosome number? Yes. How does it occur? That is the process of DNA replication. So there are so many questions. I'm not going to go through them, but make sure that you know those processes. Unless you have a question about DNA replication, about DNA code of life, let's go to meiosis. Yes, when we talk about meiosis, guys, there are some things which you need to talk about. See, this is a transfer. Okay? It makes it. You see, this is amino acid. This is transfer. We said it. This is a ribosome. This is messenger RNA. Sometimes it's up, sometimes it's down. This is what? Codons. Hey guys, how many? This question. 2 questions. The first one is how many codons in 12 bases. So remember, 3 bases is equal to 1 codon. So it means that 12 divided by 3, so makes them to be 4 codons. How many bases, how many amino acids in the 4 codons? It's gonna be 4, because each codon code for 1 amino acid. Understand? How many bases code for 5 amino acid? Remember, each amino acid is coded by 3 bases. So I'm gonna say 5 times 3, which gives me 15. You understand? So you have to know how to interpret that. And then, let's go to meiosis. Yeah, we try to explain this. They say left, right. Make sure that you translate it left, right. Ne? Yes. The first thing you need to know about meiosis are the phases, but I'm not going to go through the phases. Use IPMAT to be able to know which phase is it. But guys, something is itching me, and that itch makes people to lose marks in meiosis. Number one, identify phase in diagram 2, diagram 1, diagram 3, and diagram 2. Guys, whenever they ask you about the phase, please turn your paper if these centromere, the centrioles, they are not on the sides. So turn your paper to be like, yes, you see, it must be, I don't know, I think this is east and this is west. If I'm not a geographer, I hope this is the correct. Yeah, but it must move east to west, east to west. It must not leave what? East to west. Don't leave this. They must be in east to west so that you identify the perfect what. If it's not straight with a paper, turn it. Turn it, because if you turn it, you will be able to identify the actual what? The actual phase. So in this case, because there is space here, it means that is anaphase. But because it is single, therefore is gonna be anaphase 2. So it's anaphase 2. What about here? The chromosome, you see there in the equator. So it's gonna be metaphase 1. Yes, because the chromosomes are double, you see, therefore it's gonna be metaphase 1. If they're single, then it's gonna be meiosis 2, which is you identify the phase and then you put the word 2. That is the first thing which you need to know before you go for the paper. The second thing which you need to know about before I tell you what to focus more on this is the number of chromosomes. How many chromosomes are found in diagram 1? You will find out someone saying 46. Did they say this is a human cell? No. If they don't talk about human cell, don't talk about 46, don't talk about 23, because they didn't talk about human cell. How many do you see? 1, 2, 3, 4. It's 4. How many chromatids found in diagram 1? It's 1, 2, 3, 4, 5, 6, 7, 8. It's 8 chromatids they're talking about. How many chromosomes will be found at the end of division of diagram 2? Yes, because this is meiosis 1, therefore it's gonna be, since there are 4, therefore it's gonna be 2. Yes, that's the number of chromosomes which will be found at the end of meiosis 1. Now the question comes, how many chromosomes will be found in diagram 2 if this was a human cell? Now they said if this was a human cell. Because this is meiosis 1, human cell, therefore there will be 46, or there will be 23 pairs. Yes, I think I'm clear there. That is point number 1 about meiosis. Point number 2 is about the karyotype. Point number 2 is about the karyotype. This is where also you lose marks. Describe the structure, describe the karyotype. It's fine. You can describe that from this karyotype is for a male. If you see this is for a male, this is for a female. Because here the chromosome number 23, they have the same size and shape. While this one is big, one is small. Yes, therefore it means one is X, one is Y. But now, if they ask you to describe the karyotype, it's made up of 46 chromosomes, which are arranged in pairs. From pair number 1 to pair number 22, we call them orosomes. The 23rd pair is called gonosome. The gonosome has X and Y. If it is a male, has X and X. If it is a female, the X is longer than Y. So you describe what you see. But if they ask you how many orosomes are found in P, please guys, don't just say that is 23. Sorry, don't just say that is 40, 44, or is 22 pairs. No. Count them, because sometimes maybe chromosome number 6 we put them 3, whereby you say 1, 2, 3, 4, and you don't see that, you don't recognize. Look at chromosome number 21, but it's better to count them 1, 2, 3, 4, 5, because you'll find out. They won't just ask you a question, how many chromosomes are there? No, man, they will have to hide something. There must be a reason why they ask you that. Yes, there must be a reason why they ask you that. Maybe there is a chromosome which they have they have done the hiding, and then when you have to identify where they have hide that. Guys, give it or don't panic. Take your time, read the question 3 times exactly. If you feel like you don't understand, skip it. Yes. Why? Right? What? Start. If you see that things are not working out, start with what you think is right. I'm telling you, don't panic. Don't panic because you have not seen the diagram, Yeah. The diagram is this time they have brought it in terms of what? So why are you skipping? Skipping what? I'm not skipping anything. Yeah, I'm going to come back. I'm trying to talk about the things. After that, I come back to those questions. Ne? Yes. So guys, make sure that you concentrate on those karyotype. Make sure that you know how to identify them. Give a reason. Give one observable reason why state the gender of P. P, where is P? This one. We say that this P is what? Is a what? Is a male. Why? Because the chromosome number, the pair number 23, it has 2 different chromosomes. One is X, one is Y, or one is big, one is small, meaning that one is X, one is Y. Yes, that's number 2. That's where people fail. Number 3, non-disjunction. Non-disjunction. You see this? This is a non-disjunction question. Yes. Whereby when they ask you to which phase is it? Is anaphase. It could be anaphase 1 or anaphase 2. Yes, but looking at the diagram, you can identify that it's anaphase 1 or anaphase 2. Yes. Describe Down syndrome. How is it? How is non-disjunction lead to Down syndrome? Guys, you just talk about what is extra. That Down syndrome is as a result of non-disjunction, leading to one cell having an extra chromosome. Yes, on chromosome number 21. Yes, meaning that it's gonna have 24 chromosomes instead of 23. Yes. When this cell fertilizes a normal ovum, the resultant individual is gonna have 47 chromosomes instead of 46, leading to Down syndrome. Is it clear? Down syndrome is caused by non-disjunction. Is it clear? Either in anaphase. In anaphase, it could be anaphase 1 or anaphase 2, leading to a cell having an extra chromosome on chromosome number 21. In brackets, you can say 24 chromosomes instead of 23. Then, when this gamete, yes, fuses a normal gamete, meaning that it is having 23, it will result in 46 chromosome, a zygote with the 46 chromosome, 47 chromosome instead of 46, leading to Down syndrome. It's simple. I don't know why people make it difficult and complex and what. No, man, it's simple. Is it clear? Yes. So that is another thing where you lose marks. Number 3, give 2. Let me check here. I hope that question is here. Metaphase 1 and anaphase 1. Those ones you can do. Okay. Describe 2 ways in which this is Down syndrome. Yes. So we have answered that one. Yes. Name the organ in male that where meiosis occur. Okay. Meiosis occurs in the testes for the case of male, and then female occurs in the ovary to produce ovum. Yes. But there is something which I want where students fail. Give 2 causes of variation in meiosis. In meiosis. Guys, they are saying in meiosis. In meiosis, they're not talking about the causes of variation. No, they're saying about in meiosis. So what are you supposed to talk about here? You talk about the the random arrangement of chromosomes during metaphase. Yes, random arrangement of chromosomes during metaphase, and also talking about crossing over during prophase 1. Remember, when you talk about prophase, crossing over, you are specific to prophase 1. Prophase 1, not anything else, is prophase 1. You don't talk about something else. It's supposed to be prophase 1. That is crossing over during prophase 1, which leads to exchange of the genetic material. So please don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing anaphase. Don't go to the paper without knowing metaphase. Yes, metaphase 1, metaphase 2, anaphase 1, anaphase 2. Those we might likely to bring it. Division of a cytoplasm, what does it mean? Is called cytokinesis. This division of a cytoplasm is called cytokinesis. Lastly, on meiosis, what you can't go without knowing is know how to label. What is this? In most cases, people, they call this nuclear membrane. No, nuclear membrane cannot be seen. You see, the nuclear membrane can only be seen in telophase. Yes, yes, in telophase. And in prophase, prophase. So this is cell membrane. This is chromatid. This is centriole. The dot is centromere. This is spindle fiber. Ne? This is, what is this? This is a cytoplasm. So now I think you know. The last question whereby students get lost is draw the diagram of a cell at the end of meiosis, or draw the diagram of a cell after crossing over. Let me just show you. Let me just show you using this. Using this, I think I have the diagram here. Give the collective name, give the gender, name of observable reason, the main result of segregation, main result of segregation, describe the character of Down syndrome will result differ. Okay, sure. That's not what I want. Yes, here. Look at that diagram. Look at that diagram, guys. If they ask you to draw the cell or this after this process, guys, look at where crossing over took place. In this case, the crossing over took place up, while here is took place here and here. So if they ask you to draw this cell, maybe they are saying chromosome X and chromosome Y, whereby you say structure of chromosome Y after maybe crossing over, then you have to draw it nicely. You have to draw it nicely. Yes, you have to draw it nicely. But where did the crossing over take place? Crossing over took place on this chromatid, but halfway up. So you mean that when I'm shading all this, remember this is the black thing, so I have to shade all this. I have to shade all this. Yes, I have to shade all this to be black. And then the crossing over did not start from here, it started from here. So this variation of shading will give you a mark. Writing a caption will give you a mark. And then don't forget to label. Even if they have not asked you to label, label. Even if you don't ask you to label, at least have 2. Because in most cases, when they ask you to label, at least 2 points. Yes, they say at least 2 points. You have labeled it at least 2 points. At least then you get those marks. Yeah, please don't forget to label. If they don't say, if they say draw a label diagram, or they'll say draw and then just leave it like that. Guys, make sure that you draw and label, because it's not losing you. It's not taking, they won't cross it because it's labeled. But you get a mark if you labeled, just in case, because you are not a memorandum to know which one is not going to be credited or is not going to be, is going to be credited. So you just write. Lastly, before I forget, if they ask you to describe, write everything you know in the head. At least for us, we look for what is correct for you. If they say name, that one you only name. You don't give alternatives. But if they say describe, just write everything you know. Write everything you know. Or sometimes I call it write all the rubbish. I'm not saying that write the rubbish, but write everything, whatever you have in the head. Don't leave it, because when you leave it, you come out of the paper and say, I thought about it. I thought about it. I thought about it. No, don't do the regrets, because this is your last time to change this subject, the mark. So just write everything. If the correct answer is there, we look for it and we'll give you a tick. Yes. Yeah. Do you have any question, guys? Comparative anatomy. Someone saying comparative anatomy. Okay. Thank you. Guys, if you have any question, you let me know, because I'm here to make sure that you get a distinction. You get a what? A distinction. Distinction. Yeah. Make sure that the distinction clicks, because we want it and we will get it. You will get it. Yes. God willing, you know me, when in my region we say, Inshallah, Inshallah, we will get a distinction. God willing, we will get a distinction. That's what it means. All right. Check this. Check this. It's simple. You guys, Meiosis, I'm winding up now. Ne? Make sure that you know how to label those diagrams. Make sure that you go when you know the process of crossing over. Make sure that you know the karyotype. Make sure that you know the prophase, anaphase, telophase. Make sure that you know when you go there, when they give you a diagram, it must be east to west. Those spindle fibers, they must be east to west, meaning that you have to turn the paper. Turn the paper. No one is going to ask you, why are you turning the paper? It's your paper. No one is going to ask you whether you turn it, whether you write upside down. Doesn't matter. Turn it so that you identify the things correctly. You understand? Yeah, these phases, they are confusing people to identify. Please don't make a mistake. It's the last time for you to identify what is it. Let's try to label this and then you see. How do you call C? Is a centromere. How do you call this? The point where crossing over takes place is chiasma. How do you call this? We call it chromatid. How do you call this? Because they are chromosomes and crossing over, we call it bivalent. Yes, we call it a bivalent or bivalent. Yes. Or you can call them, sometimes you can call them, they can correct you when you say homologous chromosomes. Yeah, but scientifically we call it a bivalent. Bivalent, ne? Or bivalent. Ne? Someone might ask me the spelling. Yes. So identify, okay, we have done that. Name two ways in which structure A and B is similar. Structure A and B. How are they similar? How are these two structures similar? They have the same size, ne? Same size and shape. Good. Number two, they have the same location of the chromosomes. Not same chromosome. They are not the same chromosome. If they're same chromosome, you'd have looked exactly the same as your father. Yes. So they have the same position, locus, ne? Same position of the genes. They have the same position of the genes. Describe the process at D. So they are looking at describe the process of crossing over 5 marks. The homologous chromosome come together. They cross over at a point called chiasma. They exchange the genetic material between two homologous chromatids, and then they separate, and then they form genetically different chromosomes. So basically, that's what you need to explain when it comes to that. I'm not going to use too much of the distinction material, but I know that you guys, you will make it. Lastly, before I go, where does meiosis occur in male? It occurs in the ovary to form ovum, testis to form spermatozoa, then in plants, ovary to form ovules, anthers to form what? Grain. That's it. Hey guys, I feel good when you guys are feeling good. Ne? Don't panic, ne? Because when you panic, you make me panic. Ne? Yes. So make sure that you don't panic. Ne? Yes. Make sure, I repeat, don't panic. You're gonna make it. Let's go to genetics. Genetics. What I'm supposed to talk about? Genetics. Genetics. So we have used 1 full hour. Remember, we said that we're gonna discuss this paper for 2 hours. Eh? Yes. So we have used 1 full hour. So let's go to the second hour, that is genetics, and 30 minutes, and then 30 minutes for evolution, and then we know that we have smashed. Guys, make sure that you get all the marks in DNA, all the marks in meiosis. Yes, guys, in genetics, we'll talk about genetics. Terminologies, you know, I'm not going to talk about the terminologies, but go when you know that a circle means a female, and then a box means a male. Is it clear? That's number 1. Number 2, when they give you a question, identify whether it's a sex linked or a non-sex linked. I repeat, identify whether it's a sex linked or a non-sex linked. How do I differentiate between the two? Sex linked, they will mention it that is sex linked disorder. Always read what is on top here. Is a sex linked disorder. Is it clear? Make sure that you read it. If they say sex linked disorder, definitely you have to use X and Y. Male, there will be X and Y. Female, there will be X and X. If it is not a sex-linked disorder, please don't use X and Y. However much they have talked about same and carabo, and you know that this is a male, this is a female. Please don't use X and Y if they have not mentioned that this is a sex-linked disorder. I think I'm clear there. Number 2. Let's do one example on sex-linked recessive allele. One example, sex-linked dominant allele. One example, non-sex-linked. The first one, identify whether it's a sex-linked or non-sex-linked. Okay, now we have identified here that is sex-linked. The second thing you need to know, guys, I repeat, and I'm putting down the pen and the mouse. I wish you could see me the way I'm begging. Please, let me come closer to the mic. Please identify whether is caused by a dominant allele or a recessive allele. I repeat, identify whether it's caused by a dominant allele or recessive allele. Because if it is caused by a recessive allele, the one who is homozygous recessive is the one who is going to be affected. If it is caused by a dominant allele, the one who is homozygous recessive is going to be normal. Let me show you using here. This diagram is saying. So it means that it's caused by a recessive allele. Ne? They said it here. Ne? So it means that the one who is shaded, they're saying that is affected with the disease. Therefore, this person is a male because the sex linked is going to be that way. While this is a female, is going to be small d, small d. Is it clear? This one is a male, is going to be capital D, Y. Okay. Question. What about if it's caused by a dominant allele? Perfect. This is caused by a dominant allele. Yes. Okay, let me first finish here so that I don't confuse you. Now, let me go to the questions and see how many generations are shown here. It's 1, it's 2, it's 3. So there are 3 generations. How many kids listen carefully. How many kids are sons of 1 and 2? How many kids are sons of 1 and 2? 0. How many kids are 1 and 2? 2. Remember that this is not a kid of this. This is just came and married this daughter. And this one came and married that daughter. Therefore, there are how many? There are 2 kids which are coming from these 2. Is it clear? You see that this thing is not linking down. Is it clear? Yes. I'm done with that. Let's go to the third question. How many family members are in this phylogenetic tree? Family members. It's 1, it's 2, 3, 4, 5, 6, 7, 8, 9, 10. They are saying family members. Is it clear? They're saying family members. Don't say that this is not a family member unless you're telling me that your father is not among the family. It's a family member. But if they say how many kids for this, then I think now it's clear you know what you're supposed to do. Yes. That's the third thing. If they say that this cross with this, produce these kids, do a genetic cross, because we are in the second generation, so it's going to be P2F2. Is it clear? If they say this, it's going to be P1F1. I think I'm clear there. Yes. So I will talk about when I go to the crosses. Then we go to the next, what you're supposed to know. Yes. Can you please do a live letter? Today I join. Can you do the live again later today? I join. No, what you can do, if the live is, start from where you have started from, and then after that, join you will play the video, ne? Yes, before. Lindo Putle. Why are you removing? I didn't see what you are saying, sir. The sound at the back. Eh, someone saying there's a sound at the back. Ah, serious, there's a sound at the back. Okay, let's go to the next question. Okay, how do I find the unknown one? I'm gonna talk about it, my daughter. Um, yeah, guys, I hope the sound at the back is removed. Ne? Let me know. Let me know. Let me know if the echo sound is removed. I don't want those echo sounds. Ne? Yeah, it must be nice. All right. Let's go to the next question. How do I identify? Okay. Now someone is asking. I'm gonna go there so that we finish this. Ne? How do I identify one? To identify one, remember you have to first identify that this is caused by a recessive allele. Yes, if it's caused by a recessive allele, so what you need to do is okay, what you need to do is, okay, this one is XX, but this person is normal. Therefore, it's going to be capital D. I don't know whether it's going to be capital D, small d, or capital D, capital D. But they produced a kid who is sick. Therefore, this kid is sick with it. Don't just say sick. Sick with what? With the disease. Ne? So automatically, one came from the father, one came from the mother. Therefore, this mother cannot be like this because it gave rise to this. You see? Whenever they ask you to identify, make sure that you look at the kids. Ne? Yes, which kind of kids they have produced. Therefore, we have identified that because this kid is small d, therefore one came from the mother, so therefore the mother is like this. Ne? Now, what about the father? Remember, this is sex-linked disorder. Yes, if one came from the mother, then definitely another one came from the father. Therefore, the second X, the first X is like this, or the X is like that. Therefore, the father is affected with that disorder. Don't just say affected. Affected with the what? HIV, sickle cell, flu, corona is affected with what? With the condition they have talked about. Don't leave the answer hanging. Ne? Please, 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 please. Don't leave the answer hanging. Then the second one. Okay, someone saying, ish. Hey guys, just give me 2 minutes. Ne? Let me check why is it I hope the sound is gone. I don't know whether. Okay. I hope the sound is gone. Yeah, you let me know. Please slow down. I hope. It's okay now. All right, let's continue, guys. Let me know if the sound is fine. I don't know because I've tried to see that everything is right. I've tried to check. Let me check. Let me check here. I've tried to see that. Yes. Let's continue and see. Hey guys, I think that sound, I don't know, because we changed the system and the system I'm using is a different one from the main one. Yes. So I had to change the stations. All right, let me just continue. I hope at least you get something. Yes. Okay. I think now you have got it correctly. How do we identify this? Yes. Now let's go to the question, the next question. Then we are finding out. Identify the phenotype of individual 2. These ones I'm going to do the questions and then we see. The phenotype of individual 2. So we have said that this person is normal. Yes, is normal. A normal male or female is a normal female, or female without C. Yeah, that disorder. Then the phenotype of 1, we have identified that this person, because they have produced a kid who is sick, and the sick is like this, therefore one recessive allele came from here, another recessive allele came from here. And remember, this is a male. This is a male. Therefore, this individual is affected with SID. And then they are saying that identify the genotype of number 3. Genotype of number 3. Yes. Remember, this person is sick. Therefore, is going to give remember, is a female. It's going to give 1 chromosome from there. But because it's normal, therefore the mother gave her this. Therefore, it's going to be that way. Because they say the genotype. They say explain how individual 5 inherited disorder. Individual 5. Individual 5 is small d. Therefore, we're going to say that individual 5 got 1 recessive allele from the mother and then 1 recessive allele from the father. No. Individual 5 is a male. Is a male. Can't be from the father. Father is normal. The individual is male. So you're gonna say that the individual 5 is a male who is sick, and then therefore obtain a recessive allele from the mother. Where is these 2 marks? You have to tell us that individual 5 is affected with this disorder and is received 1 recessive allele from the mother, who is number 4. Therefore, I think that's how you can try to explain. But now let's go to yes, the problem comes in when it is caused by a dominant allele. Okay, let's look at it. When it's caused by a dominant allele, whatever is syndrome sex linked. You see, they have talked about it, but it's caused by a dominant allele, isn't it? The diagram below shows this syndrome in the family. Okay, now we are stating how many generations are there? There are 3. 1, 2, 3. Sharp. How many females are in this phylogenetic tree or in this family? Females. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Is it clear? How many females were produced by Patty and Gabby? It's gonna be females. 1, 2. This one is not family member, not a kid of this. It's a family member, but it's not a kid of this. So it's only these ones who are the kids. I think I'm clear there. Then now we go to the next question, whereby they're asking you, they're asking you, they're saying that, they're saying that, okay, name the all right, let's find out. Remember now they're saying that this is affected, né? And then this is also affected, this is unaffected. But don't forget that this syndrome is caused by a dominant allele. Therefore, these people who are affected, they're going to be dominant. Capital D. Can it be capital D, or capital D? Sorry. X, capital D, X. Is it capital D? No, they used G. Sorry, guys. They used G. Please use the letter they have given you. Don't just use what you think is correct. Therefore, it's going to be X, capital G, X, capital G, X, small g, no, capital G, and X, small g. Yes. But now I need to find out, we have, what about this one? This one is normal. So if it's normal, therefore it's going to be small g. It's going to be small g. Therefore, this person can't be this, because this person, the kid, has a small g. Therefore, came from here. Therefore, I have to say that this individual is like this. Capital, small g. No. Capital G, small g. So that one is done. What about the female? The male. Remember, this is normal. If it's normal, therefore automatically is having small g. That's why this and this, they combine to produce a kid who is a female with the 2 recessive alleles. We are done with this. What about this? Is a female who is sick. Who is sick. Therefore, this female can't be homozygous dominant. It's going to be capital G and then small g. Why? Because one X came from here, then another G came from there. You understand? Yes. Now, how many, name the type of diagram is phylogenetic tree. They're saying how many female in this family? We talked about them. 1, 2, 3, 4, 5, 6. Yeah. Male in this generation have, how many male in this generation? Generation 1 have what is syndrome? Generation 1. Where is the generation? The first generation. Eh? The first generation. Remember, this is a generation 3. 1, 2, 3. Because they are looking for male in generation 1. So this 1, 2, 3. They're talking about generation 1. They're saying with the disease. 0. Answer is 0. Because it's caused by a dominant allele. And then they're saying that give the male in generation 1 have what is disease. Give the genotype of Gabi. Genotype of Gabi. We talked about it because they produce a kid who is not sick. Guys, please take note which one is dominant, caused by a dominant allele, which one is caused by a recessive allele. They will have to specify that is caused by a dominant allele. Is it clear? Males in okay, we're done with that. Aju and Pirusa have another 4th child. Give a phenotype of their sons. And Pirusa. Yes, so of their sons. Remember this is caused by a dominant allele. The moment it's caused by a dominant allele is easier to find. For the case of what? The son. And then remember this one, son. Ne? It's a Y, it's a Y. Therefore, because this one is normal, remember is recessive, is normal, therefore this one is going to be, both of them are going to be normal. Don't forget it's caused by a dominant allele. If it was a recessive allele, then they would have been affected. But the one who has the recessive allele is the one who is normal. And the one who has a dominant allele is the one who is affected with that disease. Is it clear? Yes, that is the only trick you need to know about that. The one is normal. Yes, the one who is normal is having a recessive allele for the dominant allele, if it's caused by a dominant allele. The one who is sick is the one who is having a dominant allele. You You understand? Yes. Explain your answer above. Why? Because they are normal. Because Anju, yeah, who is a female, is normal with the 2 recessive alleles. Therefore, it gives 1 recessive allele to the males. You understand? Yes. Therefore, they are normal. So basically, that's what we need to talk about. When you go to a cross which involves an autosome, a non-sex linked, ne? Yes. Please, even if they talk about same and carao, please don't use X and Y, ne? Yeah. If they say that this diosa is caused by allele R, so you just say R. Ne? It is homozygous. If it's heterozygous, is R, small r. You don't put them on X and Y. That is the major difference between the two. Yes. Okay, lastly, about blood group. Then after that, we talk about the cloning. How many blood groups, how many genes control blood group? How many genes control blood group? How many genes control blood group? Someone is asking me, sir, it started confusing you. Daily interesting videos. How am I confusing you? Let me know. Let me know. Huh? How am I confusing you? Oh, where are you confused? Yeah, in this video description. You're saying I'm confusing. Let me know where am I confusing you so that I can sort that out. Ne? Yeah, just tell me that maybe this question or this question. Yeah, so that I can sort it out. How many genes? Yeah, correct. Correct, guys. Correct. Thank you very much. Yes. How many genes control blood group? It's 1. It means that you guys watch my videos. Thank you very much. And I'm seeing live as Mbali, as a member. Yeah. Thank you very much, Mbali. Yes, for being a member. And I saw another member here coming. Oh, Lindy Wayne Love, thank you very much also for joining us. So you are on the team, ne? Yes. All right. Never be saying that X and X are. Oh, okay. Okay, let me explain to you what I meant there. Someone is saying that X and XR. I'm saying don't use X and. Okay, let me just look for a question here. Yes, here is a question. Ne? Mbali. I think it's Mbali, ne? Yeah. Here they're saying that this syndrome in dog is caused by a dominant allele, capital H. You see? The pedigree diagram below shows the offspring produced by 2 dogs. You understand? They're not saying that this is a sex link. You understand? Yes. How many offsprings are affected by this disease? How many offsprings? You see now? Is caused by now this one is also caused by a dominant allele. Is caused, meaning that the one who is sick, ne? Check here. Even here they are saying it that dogs with the disease and dogs without the disease. So these ones which have the disease, it means that they are going to be capital H, or can be capital H, small h. Even this one is going to be capital H. You see, is a male, but I'm not using X and Y. Capital H, or capital H, small h. Whenever there is a capital H, because it's caused by a dominant allele, this individual is going to be sick. You understand? Yes. So, however much is a male, I'm not using X and Y. You understand? I don't use that. Why? Because they didn't say it is caused by a sex link. Is it clear? Yes. So in this case, but I can identify because they say that how many individuals are affected. Okay, that one is simple. But now this one is what is not affected, meaning that this individual is gonna be small h, because it's caused by a dominant allele. Whenever you see a capital H, it means that that individual is what? Is sick. If they produce a kid here who is having these two, then automatically one came from the father and one came from the mother. Therefore, they can't be this, they can't be this. So this is like this, so that one H comes here, and then this one is like this, so that another H comes there. You understand? So when they give you a question and they don't talk about sex linked, please do not use, I repeat, do not use X and Y. Use the letters the way they have given them to you. Yes? You only use X and Y if the statement says that is caused by a recessive allele is sex linked. You see, is sex linked. Even here they said it here is sex linked. Even they gave you what to use that is carried on X chromosome. Is it clear? Yes. So this situation is caused by a dominant allele, but it can be caused by a recessive allele. So I think now I'm clear when it comes to that difference between phylogenetic tree and the pedigree. Phylogenetic tree, guys, phylogenetic tree is explaining the evolutionary relationship among the members. You understand? Evolutionary relationship, like how did you evolve? But a pedigree is describing a character in a family. Is it clear? Is describing a character in a family. Yes. Okay, guys, let's go back to blood group. I think I've got it. You have got it. I'm no longer confusing you. How many genes control blood group, guys? Is one gene. Is one gene. How many genes control blood group? Is one gene. How many alleles control blood group? One gene, and that gene is blood group. Do you have any other gene? Tell me if there's any other gene. It's one gene, blood group. Yes. But how many alleles? There are 3 alleles. Yes, that is allele for A, allele for B, and allele for O. Guys, please don't make a mistake and you say allele for A like this. This is not A. This is a Roman numeral, which means 1. Allele for B is supposed to be like this. Allele for A. Don't put at here. Don't put them, make italics. Don't say allele for O like that. What is this? We will cross it. I is supposed to be like that. Is it clear? Yes. So allele for A, allele for B, and allele for O. So there are 3 alleles. Yes. I don't want to go to the distinction material to show everything. No. We go to the next part, of which the next part they're asking you how many genotypes. Genotypes. Oh, before I go, guys, back, there's something I forgot. Before I go back, I don't know what took me off. Why are males more affected by sex-linked disorders than females? Why? Or they can say, why are males more affected by hemophilia than females? Or they can ask, why are males affected by color blindness than females? So the answer you give here, if they say 4 marks, you say that males have 1 X. Yes. If a recessive allele, for example, is capital small h, appears on X, yes, the individual is affected with the disease, whatever they have talked about. While females, they have 2X. Yes. If a recessive allele appears, for example, on 1X, it can be masked by a dominant allele on another X. Therefore, the individual is normal. Males have 1X. Yes. If a recessive allele appears on the X, the individual is affected with that disorder they are talking about. While females they have 2X. If a recessive allele appears on 1X, it can be masked by a dominant allele on another X, and the individual is normal. Sometimes they can ask you a question that why males have higher chances of becoming hemophilic than females. So guys, it's the same thing. Please, you need to know about that. Ne? Yes, you need to know about that. We go to the next question. Because they have 1X. Yes, because because they have one X. This question they can come for 3 marks, ne? Yes. Is it possible for a person of blood group A marrying a person of blood group B and they produce a child of blood group O? Is it possible? Yes, it is possible. How do I know it's possible? You look at the genetics, ne? Okay, here the question is A. Before even I go there, let me just answer this question, at least one question, so that, yeah. A man who is blood group AB and then who is heterozygous, married blood group B, and then have a children. How many alleles control blood group? They're now talking about the alleles. Yes? Gene 1 is 1. Allele, there are 3. Genotype, there are 6. Phenotype, there are 4. Why am I using 4? You have A, B, O, and AB. A, B, A, B. Do you understand what I'm saying? A, comma, B, comma, A, B, comma, and O. Those are the 4 phenotypes. But genotypes, there are 6 because A is homozygous or heterozygous. Let me show you. Let me show you the distinction material, because some people, in just talking, they might not take it, and they might take it long to understand my ideas. See, you see, here is what I'm saying from the distinction material. Blood group are as a result of multiple alleles. It's a multiple allele, yes, meaning that it has more alleles, more than 2. There are 3 or more alternative form of yes, this. There are 3. All right. So phenotypes, there are 4. 1, 2, 3, 4. You understand? And then the genotypes, there are 1, 2, 3, 4, 5, 6. That's what I say. Because this is homozygous and heterozygous, homozygous and heterozygous, codominant, and then homozygous recessive. Yes. Then they're saying that 3 different alleles, which are this, and then 4 blood groups or types, which we call the phenotypes. No, the genotype, the phenotypes. Yes. So it's A, B, O, A, B, and then O. That's what I was saying. A, comma, B, comma, A, B, comma, and O. So that you understand. Yes. So now we go back to our question. Describe the type of dominance that occur in the inheritance of blood group of woman B. Describe the type of dominance that occurs in the inheritance of blood group B. Now they're saying inheritance of blood group B. Check. They're saying that a man with blood group AB and a woman who is heterozygous for blood group, yes, heterozygous for blood group B, meaning that this lady is, it looks like this. See? Yeah, meaning that obtained one dominant allele from the father or mother and another recessive allele from the father or the mother. So it can come either from the father or mother for A, and this either father or mother, because they are not specific in that regard. Yes. Use the genetic cross to show the possible genotypes of the phenotype of their children. So you guys here, this principle works to all of them. Start with P1, F1. Please, F1. Don't just say P1 and then you leave, you finish after you say F1. You will forget this F1 and then you lose a mark. So you say P1, F1, at least we shall come here and give you a T. Is it clear? Then you say P1 phenotype, because P means parent phenotype. Ne? Yes, you tell us. The phenotype is always obtained from the question. Check here. You use, because these 3 marks, it is 6 marks. 3 marks are mahala. You understand? Use the genetic cross to show the possible genotype of phenotype of what? But what did they say? They said a man who is blood group. So you say a man of blood group AB times a woman of blood group B. You don't need to tell us heterozygous, because when you look at someone, you can't say that he's heterozygous. We just say heterozygous, homozygous, so that you know what you're going to cross. So you're saying P1, F1, we give you a tick. Now you tell us phenotype, you tell us a man of blood group AB times a lady of blood group B. That's all. Then we give you another tick. Why? Because you have identified it, but you always get it from the question. So that's the second mark for Mahala. Then you talk about genotype. If you don't know the genotype, if the space, talk about meiosis. If you don't know the meiosis, if the space, talk about gametes. If you don't know the gametes, leave the space. Then you talk about fertilization. Don't know this fertilization, leave the space. But you shall come here and give you a mark there. So you have got 1 mark, 2 marks, 3 marks. Meiosis and fertilization, they must go together. If you know now, that's where you're going to obtain the other 3 marks. To know the genotype, because most of the cases, you people, you get lost on the genotype. When you get the genotype wrong, the remaining thing here becomes what? Becomes wrong. Someone is asking me, please, I answered that question. You have to rewind the video. Yes, I answered that question. Eh? Yes, for case of the DNA changing the triplet. Yes, I answered that question very well. Please, you have to watch it maybe when I'm done with this session. All right, guys, let's go to the next question. That is dihybrid cross. Then I show you what you do or what you focus on dihybrid cross. Dihybrid cross, we are not going to ask you to cross. No. What we might ask you about is identify the genotypes of the parent, the phenotypes, and the gametes. Ne? Yes. So guys, you need to know first identify which one is which. Ne? But now if they ask you what is the genotype of the parent here, for example, ne? Don't repeat the letters before you finish. For example, the genotype of this can be capital P, even capital L, then another one is capital small p, small L. So the one which are the same, you combine them. Capital P, capital L, small l. So this becomes the genotype of the parent. I'm just trying to show you how do you identify. And if they ask you to identify the gametes from here, then it still is fine. You will say that P goes with L. P, capital L. So this one is done. Now P again with the small l, capital P, small l. So it's done. This with this now, small p, capital L. Then this with this, small p, small l. Sometimes people, what they do is not a cross. It's not a cross, but what they do this. They do this, capital P, small p, capital P, small p, capital L, small l. They just cross with this, capital P, capital L. They cross this now again with this, capital P, small l. And now they cross this with this, small p, capital L. And then now this with that becomes small p, small l. See, they form the same gametes. You understand? Yes. So at least you have done. Now they can ask you, they have given you the gametes. They want you to write the genotype of the parent. Yeah, that's where the story comes from. So you start with you start with one letter, capital P. Do I have any other capital letter? Yes. So you first leave it. Ne? Do I have a small letter of the same type? Small p? Yes. If I have a small p, I write the small p there. So they are supposed to be 2 letters. But if I don't have a small p, then definitely I will have to write a capital letter. Is it clear? I will have to write a capital letter here. But because I have a small p, therefore I will have to write the small p. Then I go to L, capital L. Do I have another capital L? Yes. Do I have a small l? Yes, I have a small l. Therefore, it's going to go with small l. Therefore, this becomes the genotype of the parent. Check. It's the same thing. Capital P, small p, capital L, small l. Then when you come here, it's capital P, small p, capital large L, or capital L, small l. So that's how you can identify that. Difference between transcription and DNA replication, I talked about it, guys. I talked about it. Just wind up. I know that you will find everything you have asked me. Ne? You are trying to ask me. Yes. Lindo, yes, I talked about it. Difference between transcription and DNA replication. Yes. And even the similarities also talked about then that both the DNA double helix unwind, they talk about both there is unzipping. Yes, when you talk about differences, you can talk about DNA is formed, RNA is formed, two templates are formed, one template is formed. So you guys, we talked about it, né? Yes. All right, let's continue to the next one. How many genes control? Once you see the dihybrid, then there are two what? How many genes? There are two genes, né? Dihybrid is controlled by two genes, né? Yes. How many alleles? Yeah, in most cases, there are 4 alleles. Yes, you'll find out the capital letter and the small letter. So depending on whether you see there here, it's 1, 2, 3, 4. But there are two genes. Genes for the number of seeds per pod and then the shape of the what? The leaf. So guys, the hybrid is gonna be is simple, né? Simple. Just make sure that you do it, né? Yes. So it's the same thing here. No one is gonna ask you to cross. Before I leave this point, I wanna ask you a question. Is it conclusive to use blood group to identify the paternity, the paternity of the individual? Is it conclusive? I want yes or no. Is it conclusive? Then once I see the answer, then I will give the reason. If the answer is correct, I will give a reason why. Three factors of, okay, the experiment, that's one you must know, guys. Lastly, let's go to, before I go to, please read about the stem cells, read about the cloning. Yes, cloning, the process of cloning. I designated a full video on cloning. Ne? Stem cells, what are stem cells? The cells which have ability to modify and form other body structures. Yeah, basically you just need to read this and the examples of these stem cells. Yes, let me just show you here from Excel and Life Science Distinction Material. Yeah, see sources of bone marrow, umbilical cord, adipose tissues, amniotic fluid, uses, yeah, to generate new or replace the worn out cell or damaged cells, or to generate new cells, ne? Be used for research purposes. Yeah, so cloning, please know how to use this cloning, but most especially, let me just talk about, yeah, it can come like this or it can come as a doll. What? Do you know that this is the name of a person? Is a singer. Dolly is a singer from, I think it's from America. Do you know that? That is a singer from America. You just listen to people's songs, but you don't know that they're asking you questions and you don't know. I hope he's a singer, eh? I'm not a fan of. Okay. Is it conclusive? Okay, thank you very much, guys. Everyone is saying, sir, can you please touch on stem cells? Okay, sure. Someone is yes, I'm saying that. No. Yes. It's no. It's not conclusive because many people share the same type of bloody group. Therefore, it's not conclusive. It's not conclusive. Yes. It's not conclusive. Someone say, yes, I'm going to talk about them. Let me just talk about stem cells. Then we shall go to, not stem cells. I'm done with stem cells. We talk about the cloning. Guys, this is what I want to clone is this. I want to clone. Let me just do like this so that it gives me the full version. I want to clone this. Therefore, I have these two. I get ship A. If I want to clone this, I have to keep its nucleus. Is it clear? Why? Because the nucleus contains the genetic material. So I keep the nucleus, I throw away the shell. Then this one, I remove the nucleus because I don't want its genetic material, but I use the gamete, the ovum. But here I just get any cell from anywhere else apart from a gamete. So I throw away the nucleus because I don't want it, but now I keep this. But why do I need to have, why do I have to be with a casing of the cell? Why should I be with the casing? Yes, it's like you drinking water in a bottle of alcohol. What are we going to say? We shall say that you're drinking alcohol, Baba. Yes, that's what you're going to say, that you're drinking alcohol. Yes, Why? Because you are putting water in a bottle of alcohol. So when I put this nucleus into a cell, yes, this cell, it will look like a gamete. Remember, this is a diploid cell from anything else. So if I put this nucleus to this, then this one is going to look like a zygote, because this is a casing of an ovum, and I've put a nucleus which is 2n. You understand? Therefore, it's going to look like 2n. So if they ask you how many chromosomes are here, these are, if they say the sheep has 24, therefore it's going to be 24, or it's 2n. Yes, you understand? Yes. So now, we are saying that which method do we use? We also ask you about the method. Is mild electric shock. Then you have here the foster mother here, and then we bring this, we put it in here, and then after putting it here, then this one starts to develop, develop that embryo, and then it forms the baby. This baby is exactly the same as this. You can ask you a question: why do we remove the nucleus? Why do we keep the nucleus? Because we wanted the genetic information from sheep 1. Why did we throw the nucleus away? Because we wanted the casing of the ovum. We are not interested in the genetics here. Yes. So which method we use? Mild electric shock. And then which sheep will this, which of these two sheep will this one resemble? Sheep 1. Give a reason for your answer, because we used its the nucleus, we used its nucleus or the genetic material to produce the sheep. Yes. So when they ask you such questions, they're not that difficult. You'll be able to answer and get it right. Paternity test, blood group. So it's the same thing we are saying. This is the question I was asking. Is it conclusive? It's not conclusive, because many have the same type of blood group. So it's not conclusive to confirm or to determine that this is, yes, the parent of the child. Guys, now we are going to